Okay, all right. Welcome everyone to the microTAStic seminar today. Somehow it's unmuted. Okay. Okay, hopefully people can hear me. Can people hear me? Yeah. Okay, so we're very excited today to be able to hear from Professor Dean Ho from the National University of Singapore. I know it's very late for him right now in Singapore. It's I think past midnight, and we're very grateful that Professor Ho is still willing to stay up late and share with us his important work on personalized therapy. As a reminder, this is the, people see, I'm sharing my screen. So people hopefully can see my screen. This is the lineup of speakers for the rest of the seminar series. And just a reminder also of Zoom etiquette, please mute yourself during the seminar. And if you have any questions, please type them in the chat and we'll go through them at the end in Q&A, okay? So now let me introduce Professor Ho. Professor Dean Ho is currently the Provost Chair Professor, Director of the Institute for Digital Medicine at the Yong Lu Lin School of Medicine, Director of the N.1 Institute for Health, and the Head of Department of Biomedical Engineering at the National University of Singapore. Professor Ho is an elected fellow of the U.S. National Academy of Inventors, American Association for the Advancement of Science, the American Institute for Medical and Biological Engineering, and the Royal Society of Chemistry. He was also recently named to the HIMSS Future 50 class of 2021 for his internationally recognized leadership in digital health. Professor Ho is also a subgroup leader in the WHO Working Group for the Regulation of AI for Health. Professor Ho has appeared in the National Geographic Channel program, Known Universe, and Channel News Asia's The Hidden Layer, Healthcare Trailblazers. His discoveries have been featured on CNN, The Economist, National Geographic, Forbes, Washington Post, NPR, and many other international news outlets. So with that, Professor Ho, we look forward to your talk.
Thank you so much. It's a privilege to be speaking to the community today. I was letting Cindy know that I was actually I arrived back into Singapore from San Francisco at about 5 in the morning today. So it's good to be saying hello to everybody where I was just at several hours ago. It's good to see old friends on screen as well. And so today I'm going to talk about a few things, some of our projects and our clinical trials that we have going on at our institutes. And then I'm going to kind of focus on how we do what we do. If you look at kind of the scale of trials that we have, I think it takes a reimagining of what multidisciplinary means to us. And so, Cindy, thanks for the kind introduction. Today, the work I'm going to be showing you comes from a few entities on campus: the Institute for Digital Medicine, N1, or the N1 Institute for Health, as well as the Department of Biomedical Engineering and Pharmacology. For those who haven't been to Singapore, it's a remarkable place. I think what's so exciting is this accessibility between all of the key stakeholders that it takes to bridge ideation with validation, with clinical validation, with deployment, implementation, sciences, and ultimately adoption. So that's what we're going to talk about today. And so what I'm going to show you right now is to discuss kind of two terms that are very often used in our ecosystem, which are impact and translation. Right? I think impact and translation mean different things to different people. And so when I took the helm of WisDM as one example, but also N1 and BME, I wanted to really sit down and define what impact means to us. And with that, we introduced the term translation. And for us, it really came down to becoming a T2 type organization. So what do these mean? So the National Academy of Medicine developed this T scale, which is the stage of where an innovation is at. And so T0 being cells, tissues, and animals, T1 meaning something has reached human stage, T2 is something has reached patients, T3 is it's cleared larger trials to become practice, and then T4 is really large scale adoption. And for WisDM, we wanted to build a program where within house, right, aside from the startups that we build who can go on to become very large entities to drive clinical validation, we wanted to make sure that we could reach a scale of impact from within our walls so that we can ultimately de-risk what we do prior to spin out. But also we wanted to ensure that our impact would be counted by the patients that we help. Of course, we publish journal papers, but we wanted to see how we could harness this extraordinary level of accessibility between our tech developers, between our regulators, between our implementation sciences and clinical community and beyond, to really reach a new level of validation. And if you look at kind of the bulk of the trials that we have going on, I think we've done pretty well at this. We have an extensive clinical program portfolio at our institutes. I wanted to note that these are all interventional studies, right? To become an interventional study, it's treatment driven. These are prospective, right? They're not observational. It takes a very different risk profile and a very different regulatory profile, as well as clinical trial design profile. And so we have a team in-house that helps us do what we do to advance our ideas into prospective clinical validation. We've got over 14 first-in-kind human trials that have emerged from our technology. I will mention in particular our digital therapeutic study. We launched that study during COVID. We had intended for that study to be done in person, but based upon circumstances, we actually digitally decentralized that trial and addressed hundreds of subjects prospectively, and I'll talk about that at the end. And our trials have also led to life-saving outcomes, which I'll talk about also. We register all of our trials. We actually have more trials. Just this last 2 weeks alone, I was in 3 SIVs, study or site initiation visits. We have more trials starting up. And we register them all because I think from an engineering perspective, it's important that when we develop, design, and launch our trials, that we put them online so that our primary, secondary endpoints can be clearly stated. And so that when we report these trials, we do so in a very transparent manner. And I think from a biomedical engineering perspective, we want to make sure that we, as a department head of BME, that we train students, graduate students, postdocs, and faculty colleagues who are prepared to run trials and report them in a comprehensive, clinically relevant manner. So we've launched a bunch of trials. How do we get there? This is just a snapshot of the people who are involved in our institute, and I owe them so much because it's just a wonderful privilege working with them. So I direct WisDM. I'm going to use this as our use case today. My deputy director is a practicing surgeon, Niam. He is also the group chief technology officer of our regional healthcare system in Singapore. He's been responsible for building essentially the AI backbone of the healthcare system and is officially launched at production scale. Poonam is my research admin, and I wanted to give her a big shout out because to launch 14 trials takes a way above and beyond commitment to partnership agreements, to clinical and regulatory documents, on top of all the grant administration she does. And so it really takes a massive team effort, and there are others on our team as well that help with this. If we look at the buckets that we have of the people on our team, I'm simplifying things here for this talk, but I'm dividing them into 2 segments. One is ideation and one is implementation. I think 30 years ago, the concept of engineers speaking to doctors was quite new. I think in present day, it's important that when we think about building new clinically relevant technologies, that we incorporate all of the other stakeholders who will be essential to ensuring that new technologies are built into clinical workflows. It's a very non-trivial process. If we look at this first one, we have ideation. These are the people who are developing the technology. We have people who work in AI and ML, such as Mihul, Ed Chow, all the way to cognitive and physical performance. We have Jason, who is an ex-commando, now turned physiology professor, all the way to Raghav, who's a renowned oncologist. We have Thomas and Chris, who work in the cognitive neurosciences space, radiation oncologists, and beyond. We have Dr. Agata, who's an absolute superstar in bridging innovation with clinical validation. But if we look over here, once we validate and develop innovations, we have to make sure that they're implementable, that they actually have a chance of making it into clinical practice. And so to do this, we have a behavioral sciences, healthcare econ, and design and user engagement team. Because when we think about the user community, the user is not always the patient, right? The user can be the doctor, the user can be the nurse, the user can be the pharmacist, allied health, or the user can ultimately be insurance. And so to those ends, the very first person I actually hired into WisDM was not an academic. He actually is from the insurance sector. He's from the payer sector. And so his knowledge, Johan's knowledge, he has a deep understanding of med tech, but he approaches innovations from a health technology assessment standpoint, where the way we prioritize our programs in the Institute is how will this innovation have a chance of not only making it into clinic, but doing so at scale, and to have payers be willing to reimburse for what we do, right? Because while that, was you really start to push downstream, that typically falls outside the walls of traditional academia. But for us, we choose to harness that ecosystem that we have with that degree of accessibility to make sure we have those discussions. We have behavioral scientists, as I mentioned. We have people from our School of Communications and New Media, Renwen. We have Bina, who's a serious games expert. We have Gekong, Sujung, who are also in the behavioral sciences space, but we also have business school professors. And they're beyond entrepreneurship. They are operations experts and analytics experts. And we recently had Dr. Matthias Egermark from Roche Global. He's a senior executive at Roche. He's a clinician. He has run large trials for both Roche Diagnostics and their pharmaceutical division. He attached with us for 10 months as part of a lifelong learning initiative to learn digital from us, but then to teach clinical trial development to us as well. So he has actually reviewed and co-published now a number of papers with us, including revamped clinical trial protocols, which we all submit for peer review. We did publish a paper with him called Overcoming Pilotitis and how to do that. And pilotitis, for those unfamiliar, are scenarios where innovations just don't get past small scale pilot trial stage. His whole career has been built on bringing new developments in medicine to large scale adoption. And so it was a real privilege working with him. We have Brian Stone, who's the head of Department of Industrial Design, right? And so user engagement, user and design environment, those are important so that we ensure that our users continue to use what we develop. So we have quite a diverse team, and this is not everybody on the team. This is actually a snapshot of the disciplines represented. If you look at our technology portfolio, we have a number of innovations that span everything from clinical decision support to pandemic preparedness, to diagnostics, to food security, to GI health, to maternal and fetal health. And every single one of them is essentially at user stage or human stage, every single one of them, right? And it really took that team to do what we do. I will add that for, I would say, essentially every single one of these studies, we either are doing user engagement studies or we've already done user engagement studies to make sure that we speak to the end users and the workflow stakeholders so that we're already thinking about integration, right, before the validation's even done. One perfect example, which I'll get into, is food security. We have a food security platform and we meet with farmers, right? We meet with farmers to understand their concerns about integrating technology into their workflows. It's a generationally driven trade, right? It's not so simple just to say, look, use some AI, it'll help. It's not that simple. And that's why we engage in those conversations. So rough outline of what we're gonna do. Again, this is just kind of like an outline of going over some use cases and then talking about where we go next. So I note that the community listening today comes from diverse disciplinary backgrounds. And I'm going to focus a bit more on the individualized healthcare space. And as we know, the fields of precision and personalized medicine have been talked about for a long time, super popular. I'll talk about what it means for us and how we define precision and personalized medicine. So a very popular phrase in this field, in these fields are, people want to deliver the right drug at the right dose at the right time, right? But for each of these segments, we have to counterpose important questions, which is, if we're going to deliver the right drug, how do we pick the right drugs, right? We have to make sure we're actually doing that. Are we going to be basing the drugs off of mechanism of action? Are we going to be picking the right drugs because other groups are using the right drug? How do we ensure we're delivering the right drugs to the right people? The next question is the right dose, but what's the right dose? Is it the highest dose? And how do we derive the dose? The other question is the right dose for a patient going to be the same for that exact patient a week later, 2 days later, 5 days later? Because we think the dose can change over time. And the other thing is, what's the right time to administer, right? And so I think it's important now with new capabilities to really home in on these capabilities to make sure we're doing the right thing, right? And so something that we're looking at now is called this digital avatar, right? And what you're looking at is essentially a data-driven, but small data-driven platform to do two key things. Discover what the right combination therapies are per person. And secondly, to ensure that these combinations are dosed longitudinally in the correct manner. There are extraordinary challenges to after validation to integrating these into clinical workflows, right? So that's something that we pay a lot of attention to. And we'll talk about how we do what we do, right? And so when we think about the fields of AI, I think there's a lot of defaulting to traditional definitions of AI. The first thing I'm going to mention to discuss what we do is that we all know that we're different from each other. Okay, that's not new. We all know patients are different from each other, but it's important to note that we're different from ourselves over time, right? And so when a person receives a certain dose, it's important to dynamically ensure that that dose is correctly and evolves alongside the actual user. Traditional AI takes large amounts of population data, trains algorithms, trains models to treat the next person that walks in the room. And while that outcome may be better than standard care, it's not optimal. And we even have to define optimal correctly as well. So what we do is we use only a patient's own data to manage only their own care, right? We don't take any population data to treat the individual, which further means that we don't use any pre-existing big data sets. We use small data. So the reality is how small? When a person walks in the room, we have no data on them. We actually have absolutely no data. We actually have to prospectively calibrate the patient, which means we give them a dose, measure the response. We need an actionable marker or an image. Give a different dose, measure the response. Give yet a different dose, measure the response, to then map out what you see here is a digital avatar, right? So it's no data to start. It's actually all prospectively driven. Why is this important? If we don't pick the right drugs for the right people and we don't dose them correctly, okay, we have the potential of having good drugs available to the user, but when you give them at the wrong dose, it appears that the drug combinations don't work. Okay, that's a huge issue, right? Because in cancer, when patients receive second line, third line, fourth line therapy, when there's segments where things appear to not work, but for a subset of these patients, these drugs actually can work. You don't even have to change the drugs, you just have to change the dose, right? And so when we have this misperception of no efficacy, patients lose out on valuable time, right? We're not trying to low dose everybody. I want to be clear. We're not trying to low dose everybody, but we have found prospectively cases where improved dosing dynamically switches non-responders to responders, all right? So why is all of this important, right? Why is it important to be able to map out the right regimens and the right doses? So this is an example of a study done by Professor Ed Chow, who's my research director at WisDM. He's an old friend. We've known each other since we were about 12 years old, actually, and now we're in the same institute together. So there was a T-cell lymphoma patient that was brought to the institute, had failed 6 lines of therapy, all right, including immunotherapy. So the patient was on the doorstep of hospice or palliative care. A sample of this patient's blood was obtained. And again, we have no data, right? We don't use any pre-treatment data, et cetera, but with the sample of their blood, we prospectively run experiments to understand and essentially crowdsource their sample, the cancer cells, to tell us what are good combos all the way to what are not good combos, so that we can have some positive and negative predictivity for clinicians to understand how the prior treatment lines have failed, and then to understand what new options they may have. So it's not just looking at high rank combos, it's really a stress test. The clinicians picked a specific combo, and what happened was the patient had a complete response, made it to transplant, and ultimately achieved remission using this approach, right? And at the same time, we were able to validate the previous combos and how they were kind of failing over time. All right, and so this is an example of why this kind of optimization in designing regimens is important, right? It's a previous study that was done looking at transplant. This is purely a dosing study, and for those who don't know transplant or are not familiar, it's if you give too much immunosuppression, there can be neurotox, seizures. If you underdose, organs can be rejected, right? And so this study was published years ago. And what you're essentially looking at, for example, is the dynamic nature of a transplant patient, right? There's a week 1, week 2, week 3, week 4, looking at interactions between tacrolimus, which is your standard immunosuppression, as well as cotrimoxazole. And you can just see over time, these transplant patients are assigned a target trough range, and you can just see that patients evolve over time. This is actually not a secret. This is well known. This is why titration is the standard of care for immunosuppression. But you'll note that between patients, there are either direct, indirect, or variable relationships between how different drugs influence the trough levels of other drugs. And so what happens is, again, people are different from each other, but they are dynamic from themselves, right? And for those of you interested in... Oh, someone muted. Sorry. Anyways, so a lot of us who are interested in drug development, synergy is a popular term, right? And it's important to note that synergy alone is not enough to define good care. Synergy is relative, and also synergy is dose dependent and it's time dependent. It can change over time, right? So I think it's really important when developing therapies to ensure that we sustain optimal care longitudinally, right? And that's why kind of this rolling optimization is important. You can see here's an example of 3 different patients on the trial, and you can just see how different people are from each other at different time points when looking at drug interactions. I will note that looking at trial endpoints, and this is a small cohort trial, it was our first in human trial, the arm that we treated was discharged about a month earlier compared to standard care. I will add that time to discharge is not a standard endpoint for transplant. And the reason is, depending where you are in the world, patients are usually out anywhere from 10 days to a year. At UCLA, where the study was done, these are high acuity patients. And so a lot of them are in the ICU for months after transplant. And of course, I think most everybody in the room understands US healthcare. So when you're getting people out of an ICU faster, you're saving the healthcare system and the patient money. But beyond that, these are immunosuppressed patients getting out of the potential of community-acquired infection scenarios in a more straightforward manner, right? And so I think this was an interesting first in human study. Oncology, not long after that, we were approached by the team of a stage 4 cancer patient who was undergoing a combination regimen, including an investigational therapy. And this patient, the therapy was working, by the way. The regimen was working, but it was very toxic. And the patient was considering quitting the clinical trial at the time. So we were able to obtain the data from the patient. And as we know in oncology, dose modification happens quite frequently, but due to tox, okay? In oncology, dose modification is toxicity driven. Why? Because they already try to give the highest dose possible to patients, right? We're never gonna push above max tolerated dose. So dose modification goes downward. When a dose is modified downward, it's usually not allowed to go back upwards. because if it's a tox-guided dose change, why would we ever let them go back up again, even if it's not near the MTD, max tolerated dose? In this case, the patient's dose had been modified enough that this avatar was able to be crafted. And if you take a bird's eye view of this plot to drive down PSA, what this tells us is a 50% dose reduction to increase efficacy, right? So all of you can imagine going to an oncologist and saying, can we drop the dose to increase efficacy, right? Naturally, the initial answer was no, we're not going to do that, right? But after looking at the data, patient consent and involvement, the dose was reduced and the lowest levels of PSA ever were observed. Over time, the dosing became dynamic to sustain treatment optimization. And so what was a big change from this study was that this demonstrated efficacy-guided dose modulation, not toxicity-guided dose modulation for oncology. And I'll show you a larger example of that in just a little bit. This is another patient that was brought to us by in Singapore who was essentially at end of the line, right? Failed every line, and every subsequent line that the patient received resulted in the patient being admitted into the hospital for liver failure. And so the doctor said this was the end. The patient's family reached out, got the approval, spoke to the doctor, figured out how to build this into this patient's workflow. This patient did not have an actionable marker. For prostate cancer, there's PSA. For colorectal, you have CEA. This patient did not have a suitable marker, but their imaging was relatively stable and dependable. And so with an ethics approval of getting a few extra images, we started calibrating the patient at variable dose with Abraxane. And very surprising to all of us, at a very low dose of Abraxane, the patient's tumor cavitated. And we actually saw this response much earlier than we thought we would. And so the patient actually responded to treatment at a very low dose. And we actually, in parallel, started running cost-saving studies, right? For some of these drugs in Singapore, at least, if you use less, you pay less. And so in this case, saving tens of thousands of dollars, we actually were delivering a better outcome for the patient, right? And in so doing, helping the healthcare system as well. So what you're looking at right now are predominantly N of 1 studies. We have since scaled the study up into a larger scale study, which is still ongoing. These are all colorectal cancer patients, and CEA is the marker. And again, these are calibrated profiles prospectively, and we are now using efficacy-guided treatment. In the prostate patient, what we noted was the patient was responding, but by dropping the dose, they responded better than before. In this particular trial, we had a patient that appeared to not respond at all to one of the lines of treatment. But again, because we had calibrated them to their max dose, and there was some potential promise of reducing the dose, what was really interesting was after reaching max dose, which didn't seem to work, we dropped the dose and the patient appeared to then flip into becoming a responder. Don't change the drugs, just change the dose. Right. And this was interesting because what this tells us is for some patients, lower doses might work better for them at the right time. So I want to reemphasize, we're not trying to low dose every cancer patient. That's not what we're saying. In fact, most patients will still need clinically established doses from the large trials. However, over time, if we're able to expose each patient to a better range of dosing, this is where we start to see examples of finding more responders. This work was actually presented on site at ASCO last year. As for those who know, it's a competitive meeting to get into for clinical oncology, and we are actually going to be giving a talk at ASCO about this next summer again. So this is a really nice demonstration of being able to really advance this platform forward through continued clinical validation. Switch gears a little bit into the pandemic. In about September of 2019, I started speaking to a couple of epidemiologists. And at the time I was working entirely in oncology. And at the time I said, look, if we have a platform that can rapidly pinpoint some combos for some ID cases, it could be useful. And so we built a larger team of Ebola combo drug developers, business school professors, like I mentioned before, who deal in operations. as well as KPMG Singapore and their health econ team. In about February, we finished a study looking at a kind of a rapid platform for combo design. We did not have access to the live SARS-CoV-2, so we used VSV. And we took the liberty to kind of spend some time discussing the importance of rapid drug combo development in the event of extraordinary circumstances like a pandemic, because at those times, healthcare systems and industry will be strained, overburdened. So basically, we have to do more with less during those scenarios. And so based upon this study, we were able to get access to the live SARS-CoV-2 virus due to a wonderful community here of collaborators. So what is IDentif.Al, right? This is a platform that essentially crowdsources live virus, right? You put the live virus down and we poke it with multiple permutations of different combinations. We let the virus tell us which combinations are good, which ones are bad, and we use that to then predict even more potent combinations, which we then further validate. What IDentif.Al isn't, is it's not a drug discovery platform. We don't use IDentif.Al to design new drugs, right? There's a lot of exciting work happening in that space, but what we say is even good drugs that are designed, whether it's with AI or with humans, you have to use those drugs properly, right? You have to integrate them properly into combinations. You have to dose them properly. And so this is why we have IDentif.Al. Beyond prioritizing combinations, we also deprioritize combinations, right? And as we've learned, clinicians are quite interested in interacting with this platform to make sure that we're able to recommend or de-recommend combos. So again, this is not purely computational, it's experimentally driven. So I'm going to run through a cartoon here to talk about really how combination therapy design needs a rethink. All right, I've been meeting with a ton of teams. In fact, I was just in San Diego meeting with the entire drug discovery community. Right now, when we think about combination design, we're really kind of looking at such a small part of that galaxy of what's possible. All right. So when we think about drug combo design, a lot of times people kind of say, oh yeah, everybody's designing combos, but in vitro doesn't mean in vivo will work, and in vivo doesn't mean human will work. We all know that, right? But we first want to start off by acknowledging that drug sensitivity in a dish often doesn't clinically translate, right? When COVID hit, people were ramping up drug doses on dish, and they were saying, hey, this drug's going to work, this drug's going to work, and none of those drugs worked. Some of them did. 99% of them did not. All right. Of course, at high concentrations, a lot of drugs can look good, right? So if you're really going to understand and take 15 drugs or 20 drugs and find what is the best possible combo ratio dose that can, prevent infection or drive tumor sizes down, I really equate it to finding a needle in a galaxy, right? Because you could be talking about 1 trillion plus possible combinations. All right. So often I get approached by collaborators that say, Dean, I've got a bunch of data, I've got millions of data points for patients that got 2 drug combos, 3 drug combos, and 4 drug combos for some type of cancer. Can you predict for me which combo I should give the next guy that walks in the room? And I tell them, I said, A, you don't really need AI for this. B, that's not really the question we're asking or should be asking with AI or digital. But sure, right? Some model will predict that somebody should get the 3 drug combo and it either will work or it won't work, right? But tons of data to drive this approach. All right. But let's make the problem more interesting, right? When people tell us, hey, Dean, can you design us a combo out of 4 drugs? We always say, why don't we do more? Okay, because there's too many potential unexpected drug interactions to overlook that we should really interrogate the galaxy here. Let's think about more drugs. The problem is in traditional drug combo development, if you take 12 drugs and you study 10 doses each, you're looking at like 1 trillion possible combos. So nobody's going to run 1 trillion combos on a plate, right? It's too many plates, too much time, too much capital. Make it even more interesting. What if drug number 5 doesn't work on its own? Right? Let's say you put drug 5 down on a dish, nothing happens in the model, disease model. We tend to forget about drug 5, right? We tend to say, look, drug 5 doesn't work. Let's never think about it again. But we don't, okay? Because we cannot believe that we're going to be capable of really interrogating all of the possible permutations to uncover all of the possible drug interactions. It's not going to happen, right? So we keep it in. So instead of amassing tons of data, okay, amassing tons of data to then make predictions, we only need a few hundred data points, and we actually talk to the biology. Put the cells, the virus down on a plate in triplicate or more. We poke it with different permutations of these combinations. Right? What I tell people is you're all sitting in a room, presumably. You've got 4 walls, a floor, and a ceiling. All right. Fill the entire room up with marbles. And each marble is a permutation of a combination. Drug 1 and 6 at a specific dose. Another marble is drug 1, 8, and 9 at a specific dose. Fill the entire room up. That's your galaxy. You know, the optimal combo might be hiding in the upper right corner of your room. But in traditional drug development, we don't even look for the galaxy. We don't even know the boundaries of the galaxy. We're not even looking for it. We're using mechanism of action to pick some drugs, to poke biology, and hope the outcome is better. Traditional AI says, well, let's just get a ton of data. Let's get 5 million data points and see if we can predict better. But here's the challenge. If you take 5 million data points from a bunch of people, and they're getting a specific combination at a fixed dose, that's not 5 million marbles. That's 1 marble, okay? A fixed dose combo permutation, no matter from how many people you get that combination data from, that's 1 marble. That doesn't even let you interrogate a fraction of that galaxy, all right? So what you have to do is kind of use different permutations to populate the galaxy from which you can solve for that digital avatar, which we talked about. I'm going to go into how that actually happens. And time and time again, okay, you remember I kicked out drug 5. Time and time again, drugs that appear to not work on their own do end up in very highly ranked combinations. So too often are we kicking drugs out of consideration that can in fact potentially be useful. All right, and so I'm not going to run through this in great detail, but we developed Identify, we scaled it up, incorporated the clinical community, did our crowdsourcing. We ended up working with wild type virus, beta variant, Delta variant, Omicron variant, and we were able to develop a number of interesting combinations, some of which resulted in approved clinical protocols to trial based upon Identify findings. All right, I'm going to talk about kind of how that interaction was like. And we've, of course, since moved on to antimicrobial resistance, NTM, Klebsiella, and others. There was a recent report suggesting that by 2050, antimicrobial resistance will kill as many people as cancer by 2050. So we have to really think about better ways to address AMR instead of just simple drug sensitivity tests. Right. So far up to this point, you've all seen a lot of these smooth curves. Okay, what's going on, right? What's actually happening? So as I talked about in the previous slide, we want to crowdsource the biology to kind of feed back to us what permutations work well, which doses work well, right? And what we're essentially looking at here is these smooth surfaces that tell us that, you know, that there's a second order response that emerges, right? It's an implicit function. It's an implicit property of the biology that can be teased out by not more data, but by the right data, right? In this particular case, it's important to design the right experiments, even with DOE-based approaches, from which these second order responses can be derived. Whether it's dosing, whether it's combinatorial regimens, this has served as a principle. And people then will say, how do we know? How do you know it's an optimum? The thing is, when we run our experiments, we are getting real, right, from our crowdsourcing of biology or from our patients as we calibrate them. And when we look at some of these combinatorial derivations, we're able to implicitly look controls within these experiments. So we actually are able to know how does this combo fare against what's given clinically. And then what we do is we re-optimize and validate again. I'll give you some examples, all right? So for COVID, we've been running tons of drug combinations continuously. And when we derive new combinations, we will go and search clinicaltrials.gov to see if these combos have been studied before, either in the literature or in clinic. And what we do is we're now able to independently validate the accuracy of our approach with clinical outcomes, but we don't need any of the data from the clinical trials. It's totally independent. And we're able to do what we do within 2 to 3 weeks, not 6 months to a year, right? When pandemics hit, you do not want to spend a year trying to find out if a combo is going to fail, right? And so when we started engaging the clinicians early, we've now eventually started getting clinicians calling us saying, hey, can you rerun Identify with this new drug candidate? Can you check what combos might be possible with this drug that I'm interested in? Which drug appeared in the most number of highly ranked combos? Which drug appeared in the least number of combos in general? And by the way, there's this one trial that failed clinical trials a couple of weeks ago. How did that fare in your list, right? So this has become an actionable platform, right? And so beyond COVID, that's why it's led to AMR and other applications. So where do we go from here? As I mentioned, we've had trials approved off of what we do. We have actually had trials and IRBs approved off of the platform in general. We've put some of our findings online as an interactive drug combo design platform, just based off of our actual data. And for those interested, we even made a comic book, by the way. We made a comic book with leading clinicians and education experts. We actually, even though it's Vinny the virus is our villain, we actually started this project over 4 years ago, before COVID. We've gotten a lot of enthusiasm from this. It's in every library in Singapore. And shockingly, I went online the other day and found that one of our biggest library branches, someone actually checked this book out. I'm amazed. For anybody who wants a free copy, go to the website, you can download it. A lot of the findings here and the technology will be featured in a WHO report on regulating AI for health as a real world use case. And of course, it's been backed by a number of philanthropic societies. So I'm going to move to my last use case here, digital therapeutics. This is something that's a bit less common in the academic space. Digital therapy, for those who are unfamiliar, is not the use of digital to design drugs. Digital therapy is when the software itself is the treatment, right? And so why are we doing this? Well, in many societies, including Singapore and in Asia in particular, healthcare costs are going up, but societies are aging at an unprecedented rate. And one way to counter these challenges is to bring more care to one's home. And so when you have a digital therapy, right, what you're looking at is actually a NASA-developed game, we can actually use this to remotely do cognitive training. And we're also able to remotely monitor the trajectory of users over time. What you're looking at here, I call this a combination therapy, right? But there's no pill, right? It's 3 games. NASA developed this not as a flight simulator, but as a multitasking tool. What we did, it's open source, is that we took our CURATE platform, our dynamic dosing approach, and instead of dynamically modulating the dose, we are dynamically modulating the difficulty of each module. And from these, we are able to tease out over time personalized profiles for users. And we can do it totally remotely. It's done on the back end. It can be done on a tablet now. And we had to do this remotely due to COVID, right? And we actually brought in a former Lucasfilm animator to then work with our users to develop a more playable form of the game. And I really owe a lot to our DTX team for getting this done. We now have trials for healthy subjects. We have trials for brain cancer patients, and we have trials for seniors on the way, right? And I'll report the data next time, but we run hundreds of subjects on this trial. Suffice it to say that the outcomes are very promising. In fact, the outcomes are quite positive in terms of being able to retain improved cognitive performance longer compared to traditional training methods of fixed and adaptive intensity. All right, and so a lot of times people ask us, right? So far I've shown you on some oncology trials. We're looking at bringing ID into the clinic, and now digital therapy, which hundreds of people have been engaged with. How is it that we're able to recruit our trials and get the trials going? And with that, I really acknowledge our implementation team. Okay, we have to understand the users we aim to help. And so when you look at Agata, Vivian, right, Wei Ying, Nian, and others on our team, Smriti, et cetera, Gekong, they are experts at engaging with the users, who could be patients, who can be caregivers, who can be doctors, nurses, pharmacists. These user engagement studies help us gauge acceptability and workflow integration, right? By having these, and these are real studies, they're published qualitative studies, mixed methods that allow us to really understand the user so that we can develop solutions that have a chance of making it into workflows. And so if you look at one of our trials, this is for the brain cancer patients, right? Of using the digital therapy, we learn a lot from them, right? We assess the rationale for why they want to do it, the game itself, if it needs improvement. A lot of the patients are doing this for various reasons, right? Some of them do this so that they can help future patients. Some of them are doing this because they want to rejoin society. And so our user engagement team is now attached to every clinical study that we do. Every technology validation study we have, a user engagement team assesses in parallel off of a separate IRB, by the way, right? So that we know that our technical findings will be catalytic in concert with the user engagement data to make sure we really do have a chance to reach scale. All right, and so I kind of given a whirlwind tour of some of our clinical studies today. These are some of the conclusions, right? Like, personalized precision medicine is different thing to different people. For some people, it's taking genomic mutations, molecular alterations, and picking drugs off of that. And that's exciting work, but we don't work in that space. We do partner with that space to help recommend drugs from which we can make combos or optimize dosing. When thinking about digital, right, it's not just about how much data one has, but it's how the data is acquired, especially for intervention, for treatment. It's about getting the right data. Personalizing medicine may be dynamic, right? The right dose today may be a different dose tomorrow, right? It's important to assess users or patients in a longitudinal manner. We've validated multiple use cases. We have larger trials en route based upon the studies we've done, but the digital therapeutics is already at large scale, multi-arm, right? So what's next? What's next? Why am I showing you a photo of a plant? So what can we learn from healthcare to rethink planetary health? All right, so what you're looking at is a study we did, right? Everything I've shown you thus far is, can we optimize combinations to suppress? Can you optimize combos to suppress cancer? Can you optimize dosing to suppress cancer? Can you optimize combos to suppress SARS-CoV-2? Can you optimize combos to go the other direction, to go upward? So we ran a study where we bought a bunch of these click and grows. And what I like about them is there's almost like little wells, right? Separate wells. It's much like the crowdsourcing I'm talking about. And this is red spinach. We picked spinach first because it grows quickly and it grows uniformly. It's dependable. What we did was to design combinations of material, of growth factors in peat moss that could increase the yield of spinach. All right. And so from this initial POC, we're effectively trying different permutations of combinations in these wells, predicting out good combos, picking combos that we like, revalidating them again to make sure that the predictions are robust. And what we found was a 30% increase in yield from this study. For those interested, increasing yield alone is insufficient to really help the planet with food security, because as we know, global warming is depleting our soil. So the nutritional content is actually going down in the food that we grow. And so we actually had an independent outside analytical lab analyze our sample. And so at the end of the day, using less reagent, we were increasing yield with no adverse effect on nutritional content. We actually took some of these combos and just planted them into other types of crop with controls. And we started increasing yields 200%. All right, so this is promising. I will add that this is all done indoor, right? If we're really good, and I've spoken with foreign policy leaders, et cetera, to really talk about food security on a global scale. I will add that we're still a ways away. Okay? I have to be quite candid, because to really move the needle for global food security, we need outdoor farming, which is not as stable. I talk about user engagement. We need to train farmers. We need to engage farmers. So there's a long way to go, all right, before we get there. But as I've mentioned, we've started engaging the communities. And one thing that I think is going to happen is I suspect one of the more useful applications of optimization of combinations may be the soil regeneration space. And the reason I say that is because farmers are telling us this. We met with farmers who in the beginning didn't even want to talk to us. Okay, they were like, who do you guys think you are to come in? No, I'm not going to use nanotechnology. No, I'm not going to use sensors and drones. I can't afford it. After discussions with them, the ideas are now emerging from them on how we can possibly help. And so that's why user engagement is absolutely essential. All right, so a couple quick ending slides for WisDM in and of itself. We're housed in Singapore, right? It's a quick 18-hour, 17.5-hour flight from Singapore to the Bay Area. And so we've launched a number of initiatives to engage with the global community. As I've mentioned, we've got a couple of WHO reports coming out. The one that I co-chaired is going to come out probably in a few months on regulatory considerations of AI for health. We are about to release, I think, the clinical evaluation report for WHO for AI for health. We will be at ASCO 2023 presenting our recent trial results. So for those interested, please come and say hello. It's going to be a 7 a.m. talk, I think. So it's going to be early. We just had the World One Health Congress in Singapore. So this was to highlight some of our pandemic preparedness work, remarks from Dr. Tedros, the top people in WHO, Sir Jeremy Farrar, formerly of Welcome, now of WHO, everybody there. We actually were all in the same session. So it's been really fun engaging in a global dialogue on pandemic preparedness. The last thing I'm going to mention is to retrack our institute, right? We've got at least 14 interventional trials on the way now or completed. And our goal now is to work with as many other institutes to help, if needed, develop frameworks to accelerate rigorous and safe innovations to patients, especially interventional. And so me and 2 other colleagues recently signed with an agency, and we're releasing a book in October of this year called Medicine Without Meds. This book is not a traditional academic textbook. It has actually virtually no data in it. It's aside from our own insights, it provides a framework for how to build user-centric digital innovations and how to get them to patients safely and through rigorous validation. It has insights from venture capitalists, from patients themselves, from clinicians, from policymakers and beyond. For those of you who have seen the movie La La Land, our forward is written by an acclaimed investor and recording artist, D.A. Wallach, who just launched Time Bioventures with a visionary pharma executive from Novartis. So he's done the forward for us. Dr. Eddie Martucci, who's co-founder, CEO of Achille Interactive, one of the DTX companies to achieve FDA clearance. He's done the closing remarks for us. The book comes out in October 2023, just went into pre-sale yesterday, in fact, and 100% of our author royalties, minus our agent fees, are going to go to charity for this. So we've established a patient impact fund where all of our royalties will go to cover care for patients who cannot afford it, in Singapore specifically. With that, I wanted to thank all the individuals that support our work, all of our collaborators, and it's been a wonderful community. Our collaborators have included friends, family, and beyond, and our supporters. And so we're very grateful for those who encourage what we do. And with that, I wanted to thank everybody for joining us today, and happy to answer any questions. Thank you.
Great. Thank you so much, Professor Dean Ho, for sharing such an impressive body of work. There are a lot of questions, but I think I'll stop recording before we go to Q&A.